my group is interested in physical rearrangements of DNA. And sort of the three-dimensional organization of DNA is important all throughout the cell cycle as DNA is being rearranged. And in the interface, the organization of the DNA and three-dimensional organization of DNA is important for gene expression, recombination, and repair. And as cells go into cell division, then the DNA is being condensed into distinct chromosomes. In the organization of the chromosome and the separation between chromosomes is essential for the accuracy of cell division. So we're interested in how this rearrangements of DNA is done mechanically by mechanical forces that move DNA, make DNA loops, drive them around the cell, and reorganize them in 3D. So one molecule that is important in DNA rearrangements all throughout the cell cycle is cohesin. And this is the molecule which I mostly be talking about today. So cohesin was originally identified as a factor essential for cohesion. And so when cells divide, cohesin holds two sister chromatids all the way until separate. So if cohesin is depleted, you can see in this old picture that the chromosomes fall apart. And the essential ability of cohesin and cohesion is to resist the forces generated by a mitotic spindle, as shown here. Let me just take the pointer. As shown here, in building up this tension applied to sister chromosomes, which is essential for ensuring sort of accurate chromosome segregation. Now, cohesin is this ring-like shaped protein complex, which is has fairly long coiled arm domains, about 50 nanometers long, and 2 ATPase domains in the bottom. And given this shape of the cohesin complex, it has been postulated a while ago that the way it holds sister DNAs is through a mechanism something like this, where in this case, it's very visual to see how it can resist forces generated by the spindle by entrapping the two DNAs together. And then sort of later, it's been postulated that, you know, if it can do this, then it can probably do something like this and make DNA loops. And the organization of the DNA into DNA loops is now been established as one of the primary ways of organization of the DNA in interface cells. So on the left here, you can see a very old electron microscopy pictures, one of the first ones showing that DNA is being organized into loops. And on the right here, you see sort of more modern way of visualizing loop, which is through chromosome conformation capture techniques like Hi-C. And these techniques have shown that indeed cohesin is important for the formation of this DNA loops, as you can see here, because if you deplete cohesin, then all these loops which show as these dots of the diagonal of this Hi-C map disappear. And sort of in agreement with this has been shown by our groups and also others that cohesin can extrude loops on DNA. So you can see this in vitro experiment done by a postdoc in the lab, where a long piece of DNA is tethered via two ends to the surface of the glass. And a cohesin molecule, which you don't see here, you only see the DNA, attaches to DNA and moves on DNA, is kind of zipping and forming the loop. We can also visualize this cohesin, so you can see this. So this is a photo bleaching experiment where you can see cohesin bleaching in one step. So this way we can really show that cohesin sits here on the base of the loop, and then it's really only one or two cohesin molecules performing the loop extrusion. So now here today, I'm going to show you about two different pieces of work that focus on sort of force spectroscopy and physical force measurements. Both are published. So the first I'll tell you about how cohesin resists the forces generated by mitotic spindle. And this is mostly work by extraordinary PhD student in the lab, Martina, who just recently finished. And this was published last year. And then I'll tell you a little bit about how we study forces that cohesin generates when it's extruding the loops. And this is the work of a postdoc in the lab, George, some of which was also published last year. So I start with the first question about how is it that cohesin, which is supposed to be sort of dynamically binding and binding DNA, can resist these forces generated by the mitotic spindle? And to study this, we first established sort of reconstitution system. We wanted to reconstitute something that would mimic sort of the anaphase situation where you have two molecules of DNA which are held together by a cohesin. So what we started with is a glass slide to which we attach a molecule of the DNA. So this molecule, we can then bind cohesin molecule. And so we always want to verify, for us it's important to work with the single molecules, because when there are multiple ones, the interpretations become very difficult. So what we want to make sure is that there's one cohesin molecule per DNA, and to do this, we have this fluorescent tag on this cohesin molecule through which we can verify that it's there, and by measuring fluorescence determine there's only one, and then we can attach the DNA molecule, which has again a fluorophore attached to this, so then we can verify, and then there's a DNA molecule attached to cohesin. And once we have this sort of second capture of DNA, what we wanted to do sort of to mimic this tension applied to the DNA in mitosis, we attach the glass bead to the second DNA or to cohesin, as you'll see later. And then using this glass bead, what we can do is to use the optical trap, which you will hear a lot about today. And for those of you who are not familiar with the technique, the way the optical trap works is that there's a very focused laser, essentially focused to confocal spot, which when focused on the glass bead acts as a trap. So there's a high intensity of the laser beam in a very localized spot, and the bead tends to go in towards this high intensity of the field. And the trap allows us to do two things. First thing that allows us to do is just to move this bead around. And the second thing that allows us to do is to pull on it and then measure very precisely how much force we exert on this bead. So then the idea of this experiment is then pull on it, and then until something breaks, well, that's how you normally do the physics experiments, right? You press until it breaks, and then you try to understand what exactly happening, how it breaks. So hopefully we'll break cohesin and can learn something from this. So the system we've been using is this JPK optical trap, to which we then attach the turf elimination system, because we wanted, as I mentioned, to visualize individual cohesin molecules, which we assemble sort of on relatively standard flow cell. So the first thing we do is we attach a molecule of the DNA, we attach a cohesin molecule to this DNA, wash it with a little bit of salt, and when the salt is increased, what you can see is that this cohesin kind of diffuses, moves back and forth on this DNA, which you can see in this chymograph. And as you can see, it also bleaches in one step, indicating that this is really a single cohesin molecule. We can also now then add a second DNA, which has a fluorophore in it, and then we can visualize this correlated movement between cohesin and DNA. And then we can also measure the fluorescence, which you see here, that in all these experiments, all cohesins bleach in one step, and this step is exactly the same, which tells us this is really the single fluorophore that we see, verifying that we visualize single cohesin molecule. And then the experiment itself is done something like this. So you can see here on the left the movie. So the bead, and this is, in this case, it's slightly fluorescent just for the purpose of visualization. So we pull on this bead, the DNA is being stretched, and this DNA being stretched, there's a cohesin holding on this bead, as you can see on this diagram.